Hello! Oh my goodness, who are you? I'm being mauled by all of these extra people in my screen.
Very good entrance, Douglas.
Yeah.
Hi, I'm Dr. Smith.
Yeah, he's Dr. Smith.
Yeah, and I'm happy to be here. Very happy to be here. I'm in the chat, cryptically labeled Dr. Alex Smith, and I am happy to join you. I'm not sure if I would have been the Saw-Whet Owl bottom row. Doug, shh. Chill. This is Doug. Did you meet Doug already last week?
I don't think so.
You met Lily.
Yeah, Lily.
And maybe Caramel as well. Caramel's more chill back in the corner. Doug's got more energy. But great to see you in the chat. I see lots of people. How was the trip? It was fantastic. September in Costa Rica in the Northwest is rainy, but almost every day in the morning it is not rainy, which means you can get up early and go and get up to the tops of these mountains in cloud forest and look for tiny little insects and ask questions of them and say, tiny little insects, why do you live where you live? Is it because of the abiotic conditions? Yeah, so that's me in a happy place.
Yay.
Yeah.
Okay, let's check in with everyone though. We are feeling, are we filling with rage?
Sí, yo hablo español. No, nosotros hablamos español.
Sí,
yeah.
es la única idioma que nuestros hijos no hablan.
Yeah.
Así que podemos hablar sin que entiendan.
Yeah.
Okay, let's go. We are filling with rage, just like Jasmine is. I agree.
What's the least?
The least. Oh, this one here.
Top, well, that's good.
This
top left.
A1.
That's not how you want to start the week.
Okay. Yeah. Let's. Yeah. Okay. So you are going to need a ruler. I hope you've noticed this information here.
Links in the chat.
Links in the chat. How to get to one on your screen. If you don't have one, we're going to be doing some measuring. Today is going to be awesome.
Sharp07 is banding Saw-Whet's Owl this fall.
Oh, good.
That's awesome.
Yay. Wonderful. Okay, cool. I'm going to clear this now. Thank you so much for sharing with us. That's what we like to see. Here we go. And annotate, clear. And moving on. You met this guy. Happy place. Cool. So.
And thank you.
Oh.
We had lots of, like everybody does, but lots of scheduled navigation in the fall and the summer with getting up to the high Arctic and getting down to the tropics and also ensuring that our family is alive and healthy. And so thanks.
Oh, thank you.
Yeah.
Wonderful. Yay. Which hand? This one. There.
Meatball spaghetti.
Yeah. Okay. Here we go. So just a little reminder, your homework. If you haven't done it yet, no worries. But here's just some guiding questions. Feel free to share your ideas on the discussion board and talk with other folks about them and see what you come up with. And what's next? Haha. For Monday or for the near future. Let's kind of like drill in a little bit. Some of you are already there. Some of you are kind of exploring more generally these life cycles. So let's be even more precise. So coming up, either we might get there by Wednesday, but if we don't, then it'll be Monday. Again, none of these things are like super critical about timing, but rather just allowing your brain to go there when you have the opportunity, right? So some of you might have a very busy Tuesday and can't do it. So this is for you to decide on your own time when you do it. Let's drill down now to one advantage and one disadvantage of the strategies of Glaucidia, and then one advantage and one disadvantage of the strategies of the veliger larvae.
Glaucidia.
Yeah.
I have some larvae named
You'll have these slides at the end of the class, so you can go and do all of that. And also, we do have at least one note taker who has been taking notes since the beginning of the course, which is great. It's always nice to get more. So if any of you want to be a volunteer note taker, please get in touch with the course email address and we can connect you to the Student Accessibility Services office to be able to do that. But we definitely have one because I've seen the notes from the beginning, and they will be made available as soon as possible. Okay.
Y'all, you've got some great thoughts in the chat already about veligers versus glaucidia.
So good. Okay.
Not versus. Not
versus.
Like, but like alternatively.
Yes. Okay. Cool. So that's for today. And again, you know, you can go through them and read them, but use them as a bit of a checklist, right? Okay.
Today's fun. Today's a lot of fun. Like we're going to harvest some measurements, and we're going to talk about, I think, one of our favorite things in terms of communicating science, which is how to do it visually. Or maybe how not to.
Or let's think about how not to.
Yeah.
Just because that's really fun to kind of get our creative juices flowing.
We'll call it SciComm fail.
SciComm fail?
Yeah.
Okay, good. Here we go. So, how to display data badly.
This was a paper.
Yeah, we spend a lot of time, I mean, like, we do, spend a lot of time thinking about how to communicate our science. And how to make sure that it is well understood and that it is creative and beautiful. And it really makes such a difference. You know, just putting in a little bit of attention to your graphs or your infographics or your sort of visualization of or representation of, right? Because it doesn't necessarily have to be visual. And if we're thinking about accessibility, we have to think about non-visual ways of representing data. So what we want to do is we want to spend today kind of talking about that and playing around with it a little bit, making our own data set, reflecting a little bit on the process that we took, the methods that we took, thinking about accessibility, and also thinking about inclusion. So that's kind of what we're going to be talking about today.
Yeah, how to tell the story to the biggest audience possible.
Right, the best story to the biggest audience, right?
Can I interrupt with this just in and said, if your fire alarm is going off, don't stay with us.
Go out.
Listen to the fire alarm.
Watch the recording later if you have to.
Later.
Okay.
We'll do like previously on Biology 1070.
Yeah. And let us know if you're okay.
Yeah.
Because that matters. Here we go. So, how to display data badly. Don't do this. Now, this is obviously an extreme example of what is it?
What?
Sharpie Gate?
Sharpie Gate.
Is that what we're calling it? Yeah, Sharpie Gate. But it's really important, you know, when you make a mistake to own it rather than to add it to your data set. And this happens a lot. Like this isn't just the only case of it happening.
And in terms of so we're not saying never annotate a hypothesis with speculation. that can be actually kind of fun, and it can be a great way to brainstorm and in a presentation say, this is what we found. We thought maybe if we extended the analysis, but you're being honest about it and upfront about it and saying, I would speculate not what happened here.
Okay, good. So
don't stamp.
Don't stamp. Hold off. Just wait.
Hands off the stamp.
Hands off the stamp. Get it ready though, but don't stamp.
Stamp hover.
When we say stamp hover. When we say 3, we want you to go ahead and do that, but don't do it yet. Just wait. So let's take a look at what is wrong with this graph. Okay, and what we want you to do is to not yet. Get ready to stamp where you think. Try not to stamp like on the words so that other folks can see them. And I'll just keep on delaying a little bit so that everyone can pick one that they're going to stamp on. Are you ready? In 1, 2, 3. There we go. Well done. Super good.
I love stamping.
Woohoo!
Interesting.
Yep.
So in the chat, lots of you are choosing labeling. What's wrong with the labeling?
Yeah, what's with the labeling? What do you mean by labeling?
No labels on Axis.
Oh, that's good.
This is true. And I'm sure this is something that you, in your lived experience, people have hammered you for, which is fine.
Yep.
And good.
Super.
But some of you talked about...
Madison's doing it for us here. Nice. Really good. Good job, Madison. That's great.
Yeah, so then, no labels anywhere. Graph is not specific. No consistency in the axes.
Yeah.
y-axis goes up crazy fast, which is like in our household, it's currently called sonic fast.
Yeah, they're into Sonic, right?
Irresponsibly fast.
Yeah.
Scales are whack too. I love it.
Perfect. Okay. Yes, that's great. Typograph, some of you are like, typograph, sure. You know, there are other ways that could be represented.
Certainly, like for instance, color. Like we talked about communicating to the widest audience equitably.
Yeah.
Is this equitable to lots of people with who see red and blue in different ways? My guess is no.
Okay.
But most problematic,
Yes.
I think we would agree,
Yes.
is
Yes. The scales.
The scales.
Axes.
The axes.
Yeah.
Not so much the X. I mean,
yeah.
X is fine. Time.
Okay. Although, are they equal days? This is 5 days apart.
Yeah, you're right.
5 days.
Yeah.
This one's 10 days apart. Oh, the y-axis too, or the x-axis too.
Yeah.
Look at that. That's 9 days apart. Okay.
Yeah, so take it. Yes, exactly. So we replotted, or we grabbed a correct plot.
Oh, did we?
Yeah.
This is
So
what it
it's
actually
clear.
looks. Did you put it in?
Yeah.
Oh, you're amazing. Okay.
I sometimes.
Oh, this is what it would actually look like.
So in terms of the story that you're telling, what's the difference between the former, the bottom left-hand insight, and the more correct? What's the difference in the story? Your audience.
My face. So if I'm looking at the one on the right, I'm like,
Nah.
if I'm looking at the one on the left, I'm like,
Ah!
yeah,
Yeah,
Isaac.
exactly.
That's good. Good,
Yeah.
Noor. Yeah.
Better or worse scenario, increasing slope, more drastic and dramatic. Yeah.
Yeah.
Yes.
Excellent.
You guys are well done, folks. Y'all are on
the
this.
ball. Okay.
So let's change it up.
Let's change it up. Here's another one. Hold
Change
off
it
with
up.
the stamping, please, because it's so
Change
much
it up.
better if you can like stamp authentically. Okay, here we go. So again, try not to stamp on the words so that folks can still see. Take a look at this, and don't worry too much about whether or not you understand, what the splice site strength is. Like that doesn't matter. Just take a look at what's being demonstrated here. I'll just keep on stalling for a little bit so that you can look at it. And in 1, 2, 3, go!
Oh, you guys are awesome stampers.
You're really good stampers. Okay, there's like a y-axis type of graph thing going on here. That's good. I love it. Okay, really good. So what's up with the y-axis? Tell us in the chat. Some folks are like, the x-axis is wrong. Tell us what's going on with the x-axis and the y-axis. For a second first, if you don't mind.
Throw it in the chat.
Oh,
Y-axis is
Maddie and Nora
inconsistent.
showing us. Oh, look at that. That's really good.
Yeah.
Cool. Wonderful. Well done. Okay, type of graph. Yeah. What type of graph would you prefer to see?
Not consistent. The y-axis is not consistent. Doesn't look like the graph corresponds to the y-axis.
Yeah.
So think about kind of the style of graph.
Yeah. Somebody's circling going, what? Okay. Take a look at this because...
Oh, yes. S. Davis 22. A bar graph?
A bar
question
graph.
mark, something like that. Something that's discontinuous.
Right.
Right? Because by joining these points, you're saying this to that leads to that. And it's like, no.
So let's put this into words. Basically, what this graph is showing is that as you move from being a rhesus monkey to a gibbon, to a chimpanzee, to a human. So like the same organism is moving to be different species.
Yeah. Tell me about your experimental methodology.
I mean, this would be an excellent plot for a movie, but it is not. These are not continuous data. These are discrete data. Okay, super.
They keep their secrets.
Wahahahaha.
Is that all the examples? No!
No!
We have more!
We have
I
one
love this
more.
one. Now stop. You're going to want to hammer on this one.
This one's amazing. So we'll just give you a few seconds to take a look. A few more than a few seconds. Hold off, hold off. And in 1, 2, 3, go! You're awesome. All of you are awesome. Super good. Okay.
It's kind of a competition, eh? Between
Yeah,
type
yeah,
of graph
yeah.
and incorrect
Right.
inference.
So why type of graph? What type of graph would you
Yeah.
want to see? I hadn't ever thought about type of
Look
graph.
at that phrase.
There we go. Love it. Hodgkin V.
Hodgkin V. Causation does not equal correlation.
Very good.
How do the axes relate? And in Wolfers, yes.
Yeah. That's
correlation.
Really good.
We usually flip that phrase. Correlation does not equal causation.
Yeah.
But,
yeah. Well done.
Would if you could, eh? To say, like, we could talk to the National Science Council in Canada and say, we've had some Nobel Prize laureates. We need some more. Now, to identify this, we're going to eat more chocolate. Yeah.
Now, it is possible that chocolate consumption is related to socioeconomic status.
Yes.
Right? And gross product or whatever, domestic product of a country.
Affluence.
Right.
Yep.
And so if there are lots of people eating chocolate, then there might be lots of investment in science, which means that there might be more Nobel laureates, but you wouldn't put chocolate on your x-axis. It is not causal, right? Good. Fun. Okay.
Sounds like some of you in LA are coming back from the fire brigade and fire drill. So welcome back.
Welcome back.
What you've missed is going to be on the YouTubes later on, and
we're just warming up.
It's fun, but we're just warming up. We have not measured anything. We're still talking about visual data presentation.
That's right.
So this example is not a video, but it's a screen grab from a Fox News broadcast of about 7 or 8 years ago, where at that point in 2014, they were talking about the fact, they were saying, Al Gore 7 years ago in his Incontrovertible Truth said that the Arctic would be ice-free in 7 years. So they've taken two plots here, and they said, look, in 2012, there was that much ice, and now there's more. So climate change, not a big deal. The ice is actually increasing. But this is the data. So use the stamp tool to select the two images that Fox News used to compare, and then think about what the problem is. And on the next slide, I think we've got yeah.
There we go.
So you can put in the chat. Yes, there you. I see it in the chat already. Spears A. One is the summer and the other is the winter. Is that a reasonable comparison?
No.
So, fraudulent presentation.
Fraudulent presentation, right?
Yeah.
Totally seemingly decent data, totally fraudulent. Yeah. So this is important. We see a lot of this, and the ability to critically think and to be able to see through these things, it takes a lot of work. Yeah. I mean, we've all been through this over the last 2 years, right? Yeah. We are tired with having to constantly critically evaluate these data, but it's really important to understand how leadership or how politicians or how university presidents are using information in order to make decisions about our community, right? Yeah. Or about our planet.
Yeah.
So what we want to do...
So this is true for your day-to-day life. You have lived it. It is part of our lived experience, our shared lived experience now. But it's also true in how we do science in sometimes less topical or virus or community health based. Like, for instance, in terms of measuring phenotypic variation. In this case, with our example today,
in humans. Okay, so let's take a look at some phenotypic variation, right? So when we talk about
Wait, did we talk about the phenotype? Do we know what a phenotype is?
Well, I think we know what a phenotype. Do you know what a phenotype is? You're like,
phenotype?
No.
Phenotype?
Maybe not.
Phenotype?
Yeah. Oh, yeah. Nora's like, yeah, we covered that. Okay, cool. Everyone's like, yeah, we're good.
Physical characteristics.
There we go.
Fernand Z.
Good.
Yes.
Well
it was in pre-lecture readings. Good. But did you do them?
Yeah. Okay. So when we talk about phenotypic variation, we talk about usually we're talking about intra-species, within the species, right? There's all sorts of delightful variation that basically allows for me to not look like this guy right here, right? We are different. We are different heights, for example. That would be a phenotype that has variation within it. What I want you to do, though, and when we think about critical thinking, I just said that, there's phenotypic variation within traits like height. Think about traits for which there would not be any variation. Yeah. Okay, so think about a trait that has no variation as we talk about traits that do. So one of the traits that we can talk about is height, and an excellent kind of example of that or demonstration is the variation that existed between Manute Bol and Muggsy Bogues. Both of them played their was it their inaugural year together?
Yes,
Yeah.
They had overlap on one team. And
On one
certainly
team.
overlap in the league.
For one season where they actually played together. So Manute was 7'7". He's passed away. He was an activist from Sudan who was fantastic at getting the ball in the net. Not so good on the court. Muggsy, though, was brilliant. Basically just went under all of the other players.
It's his highlights you're watching here. He was an early member of an early Toronto Raptors team late in his career, but he's like a water strider. It's like
so good. 5'3". Okay, major difference in phenotypic variation between the two of them.
Somebody in class is 5'3". This gives them hope.
Oh yay! Okay, so if we take a look at height in humans and we present it badly, this is what it might actually look like. So a whole bunch of people were told to wear different clothing according to their assigned sex at birth. And so those who were assigned female were told to wear white. Those who were assigned male were told to wear black and to stand in line on a football field behind their different height category, okay? And what we see here depicted is what's called a normal distribution. And this is a term that doesn't just mean like your average distribution. What we're talking about is something very specific. A normal distribution is a bell curve where there is like equal distribution on either side of the mean of the average. Okay, and you can Google all of this stuff later and take a look. You get this bell curve, and it's pretty bell curvy. Maya is 5'2", we now know. And anyway, so these are poorly represented data, and if you want to share why you think they are poorly represented, please go ahead. But they really don't allow us, thanks Chris, they really don't allow us to appreciate any difference between those assigned male and those assigned female at birth, right? We can't really compare these in any kind of meaningful way. So there are better ways of representing these data. Here, I just need to close that. And so is this a good graph as you're going through your thinking, your studying, right? See if you can come up with all of the reasons for why this graph really sucks. We could look at it this way, perhaps, if we wanted to. And what we'd love for you to do is to take a look at these data while you're studying and preparing for any sort of future, topic test or something like this, is to come up with like 2 or 3 conclusions, statements that you can get from these data. Okay, and we're going to do that now on another set of data, so we'll show you how to do that. Okay, cool. Because what we're going to do is we're going to measure phenotypic variation right now in class. So get ready. This is going to be quite active. What we'd love for you to do is to not only do what we're asking you to do, but reflect on the process, because we are going to give you very clear instructions, and then we're going to, unpack those instructions. Okay, so just hold on to the way that it's going to make you feel, hold on to the questions that you have about it, and then we're going to discuss a little bit afterwards, if that's okay. So about 10 minutes, might be some tension, and then we're going to relieve that tension. What we want you to do is to take your left hand, okay? That one, right? Okay, good.
That one, correct.
That one like this. Here, there's my left hand. There's your left hand. Oh, look, we have the same wedding rings. What does that mean? Yeah, we haven't told them yet. I'm sure they know. We're married, okay? Yay! Okay, so ring finger, index finger, and what we want you to do is to measure those lengths. Okay, so hold on to that instruction, and we'll give you some more clear instructions here. Okay, so all of the links are going to be posted in the chat. Is that right? They're coming. Okay, but using a ruler, on the back of a field guide, if you have one. We used to have a field guide that was assigned to this course. Use the screen one, use any kind of ruler. Measure your index finger and your ring finger. So separately, we want you to write down or keep note of the length in millimeters. And when we ask you to share the data, we will ask you to share only the number. Don't put the unit in, okay? Just the number for each of these things. So we're gonna just get you to measure, and we'll pop in the links in the chat. And then you can go and do that. Go. There they are, based on your surname. Now, the reason why we have it separated is because Google Sheets only allows 100 people on each sheet. We don't really care whether it's by surname. Actually, we're just doing that to, spread out access. If you get onto a sheet and
use it.
Yeah, and it's closed because there are too many people on it, just pick another link. It's not a big deal at all.
I'm just trying to avoid niche competition. What's that? Ooh, check back later.
That's good.
Yeah. Yep.
Hold on to that thought.
Yep.
Okay. Again, there will be a bit of tension for just a few minutes, and then we will unpack it.
As a measure of the quality of Jasmine's work, I am still
You're still
annotated.
Like,
I am still calibrated to that beat.
It's a good beat, eh? That song was pretty good. Always happy to learn about new Canadian music by diverse artists. So please feel free to share any of your favorites. If it makes us go like this, then we will feature it in the morning. How are we doing on the, how about when you're done, give me a stamp in the bottom right-hand corner of the screen. It's working. It's working. Wait, you're gonna like this. It's really cool. So we've set it up. He's set it up so that it is generating graphs automatically based on where you're putting the data in the different cells. So we're gonna have graphs in just a second.
Nice.
Lots of you are ready. If you don't mind, we'll do that.
I do.
I know, but okay.
Let me do it then.
Okay. Lots of you are ready. This is great. Awesome. Thank you. And I think I've got to refresh my slides too for it to show, right?
Yeah, you do.
Okay. Cool. Are we ready? Are we good? Yeah. Okay. So I'm going to stop this for a second. Thank you so much.
Yeah, you're going to escape out of the
Yeah,
presentation.
I'm going to just stop the presentation for a second so that we can update the slide.
Take it away from the recording so that it's clever.
Here are the data. We play around with the recording so that we don't have too much like dead time. Okay, here we go. So what I'd like for you to do, first of all, I'll clear the annotation. Okay, we're going to do that whole thing about two conclusions. Hold on one second, though. It's still a little bit awkward. What we want you to do is to come up with two conclusions based on the data, okay? Here we go. Two totally separate conclusions based on these data, and pop them in the chat for us. Okay.
Oh, thank you for that. I'll go look for that.
Here we go. So take a look at these. What are your conclusions? Pop them in the chat.
Some ideas popping up.
Great. There we go. That's good. Good. Really good. Okay. Super. Yeah. Excellent. I mean, the other day I was looking at different maps with my son, and we were coming up with different conclusions based on the different maps, right? That we're showing. They were kind of fun maps. They were like, 50% of this country lives, south of this line kind of deal, and trying to like come up with these different conclusions, right? To kind of teach my kid how to like interpret data, right? And to kind of derive meaning and value from them, and that's by far the most important thing that we can do, rather than just say the average is this, right? What does that mean? How does that matter? How is it relevant? So this is a really important skill that we're hoping to be able to pass on to all of you. So 2 conclusions. If you want, when you're studying, come up with 3 and 4 conclusions, just to again really appreciate some of these nuances and differences. Okay, let's talk about this, because there were some problems associated with what we did.
Mm-hmm.
And we can pop them in the chat. So what patterns did we observe? We did that. The next question is critical, and already some of you have noticed it, perhaps more so than others, because you were excluded from this particular exercise. Who did we exclude from the methodology that we followed? So let's be as sort of open as possible about this from the very beginning of the exercise to right at this moment. Who did we exclude from the methodology?
Yep.
Me.
Lots of me. Yep.
Yeah.
Yep. Yes.
People with no hands, absolutely.
Non-binary people.
Yeah.
Lots of people saying non-binary. They, them, non-binary.
Yeah.
People with no hands.
Missing
those with
fingers.
No left hand, missing fingers.
Yeah. They, them. Yeah.
No access to Wi-Fi.
No access to Wi-Fi. Totally.
Yep.
Yeah.
Some people commenting on age.
Yeah.
That we're not looking at a whole set of
Yep.
people.
Very good. Yep.
Yep.
Yep. All of these things, right? Folks that aren't either male or female,
Yep.
folks that aren't men or women,
Yep.
Those are very different. It's important to understand those differences.
People without rulers.
People without rulers, very good, right? Without the opportunity to engage because they don't have the tools, right? Super important.
People who may not know how to use Google.
Very
younger
good.
And older adults.
Very good. Okay.
Yep.
You're good.
Yeah.
You've got them all. Now,
these are not trivial problems.
No.
Either. And so what might surprise you is how frequently these problems come up in the real world, in scientific data that is presented and packaged and published.
That's right. All the time. I am tired of spending, my valuable time filling out a survey, answering a question, checking non-binary, and then reading out the results that have been done where it simply says that due to low representation, all non-binary categories were removed from this analysis, right? Yeah. So it's not just whether or not they thought of asking trans or non-binary folks or any other type of folk that doesn't, fit within those categories. That's nice that they ask, but then so many studies eliminate those data, right? Because the category was just too small. We couldn't use it, right?
So that speaks to problems
after
months ago in
That's
how
right.
you designed and how you included
Yeah.
people in your survey.
Yeah. So I write and I say, if I check these boxes, am I going to be included or not? And that's, I think it's an important distinction, right? A lot of us remove categories. Like, for example, how much do we know about spiders?
Yeah. Yeah. So spiders, what we know about spiders, what you may not know about spiders is that almost all species of spider can only be identified by looking at the adult male. That is not representative of spiders that you catch or spiders that live in the world, which are generally, for all the species that I know of, have an equal sexual representation in the wild, let alone the juveniles. Right. So what we know about spiders to be true has an incredible bias towards the male, and that's because of an incredible bias that is ingrained into the taxonomic community that we have trained over the last 250 years that says, yeah, well, how are we going to identify things? Well, look at the males. And it's like,
So if you're interested in another example of this, did you know that female birds sing like beautifully? We didn't until a female scientist asked that question.
Yeah.
And it turns out that female birds do sing. And we only ever talk about the songs of the male birds, and we only record the songs of the male birds. But female birds are singing too. And these types of new revelations are really important,
Yeah.
right? We have a researcher here at the University of Guelph called Dr. Glenn Pyle, who works exclusively on female hearts and risk for cardiac disease and problems. And he's dedicated his lab and his resource to understanding half of the hearts that we actually aren't studying much of. So these types of realizations that we've been missing out on important populations is really important, and it's changing, and it's also relevant in wildlife. And I know a lot of you are interested in wildlife, but the way that we approach our science creates an incredible bias in what we collect. So we have to be able to check that bias as well.
So bias is all over the place in terms of who's recruited, who's encouraged, who's included, human biases or biases amongst us, but then also the biases like we talked about with the spiders and the birds, biases about literally how we see the world. Can you imagine, so you're a birder, lots of you are keen birders, that you had binoculars, the best new binoculars that you wanted to buy, a new spotting scope, and it only showed you the male birds.
Oh no, that's good.
That's essentially what we do for most of life, because most of life is not birds. Most of life is less than a gram, less than a centimeter, doesn't have a name. And when we do have a name, it's almost always dependent upon being able to identify only the male.
Yeah.
It's like so much work left to do.
Yep, absolutely. Cool. Thanks.
And I've been reading through your thoughts, and I've got a queued up kind of a quote here in a second, but boy, oh boy, I agree with all of them. Yep.
Wonderful.
Lots of them.
That's really good. Thank you for going through that with us. Thank you for your patience. Thank you for allowing us to collect those data and make a mistake and discuss how we made that mistake. And hopefully this will be an important lesson for many of you, and some of you it will be a reminder of some things. And for that we are sorry to have had to have reminded you. And we do see change. We are optimistic about change. Okay.
Are we going to do that?
No, let's not.
All right.
Let's move on because we're we're running out of time. Ish, right? Yeah, doggies.
Now wait a second.
Wait.
And this is another graph. Which dog is cute?
Oh.
I'm guessing cute, you were talking about the lily put.
Okay.
Yeah.
So this finger ratio thing has been done. I'm sure you've heard about this 4D, 2D thing, this ratio.
Tell us if you have.
Yeah,
because
have
to
you heard
me,
about this ratio thing?
This was novel, but boy, oh boy, the literature is just like full of this stuff.
Yeah.
Have any of you heard of the 2D, 4D?
Yeah.
No,
you heard?
no,
Yeah.
yes.
Okay.
And LOL, that's
Heard
an
of it?
appropriate scaling.
No.
So lots of no's.
Yes, some yeses.
Good,
Okay.
some yeses.
Good.
Let's talk about it.
Let's talk about this.
It's bananas.
Bananas. And when I think about it, I think about the graph about chocolate and Nobel Prizes.
I just think about chocolate.
Okay, so let's take a look at this graph, if you wouldn't mind, and see if you can come up with one conclusion. Just one.
Excel has got better since 2000.
Yeah, that's one conclusion. But what else? Let's see if we can come up with a... People are stupid. Very good. That's very good. What else? Another conclusion.
Can we phrase that in the form of a question?
So...
Nice, Hodgkin. What does sexuality have to do
with
with
this?
it?
Oh, nice.
Why? What?
You're already skipping. You don't even want to look at this. Now, for those of you in the room who are like looking at their fingers going, "Oh my goodness, you mean I'm straight?" No. No. This is a chocolate Nobel laureate kind of a thing, right? Some guy named Mark Breedlove, which I think is funny, decided to go and measure the different finger lengths of a whole bunch of different folks. Now, what I love about this...
Mark Breedlove is kind of whack.
What I love about this is that within the group that are here, your 553 people, we've almost exceeded the total number of finger lengths that Mark Breedlove measured.
In Nature.
And this was published in Nature.
This is like the biggest scientific journal in the world that people aspire to.
Right.
And a solo authored paper there is a career maker.
Yeah.
And so he has spent his career, the intervening 22 years, doubling down on this.
Yeah.
He continues to do this research. Others have.
Yeah.
There is an entire field on finger length ratios.
That's right. And folks are just like bananas for this ratio, right?
And boy, they put it to all things. It's like to athletic results in athletic competition, the likelihood of being selected or achieving marks in grades in a course, in different fields, in different susceptibility to COVID.
Yeah. Okay. So people are asking why, and it's, yeah. Okay. Let's move on to some of the official criticisms of this.
Yeah.
Because first of all, this is a terrible graph. Irrespective of like the kind of ridiculous nature of the hypotheses that this person was testing, this is also really bad data presentation. Okay. So can we focus on that for a second?
Yes.
So why is this bad data presentation, right? Going back to the beginning of class, what is wrong with this graph? Let's just accept the data for a second. What is wrong? It's hard to read.
Bar graph is
this
hard
chat
to
is
read.
so funny. Keep it up. It's great. What
hard
else?
To read.
Yeah.
Bars are all wonky.
All wonky, really good.
Yep. Patterns are really similar. Yeah, now there was literally only this long ago. I mean, it's your lifetime, I suppose, but it's not so long ago in the scientific literature where journals required you to submit not color versions. Like you could not submit a color version of your figure. Even that, though, I think your criticism is still valid, that these patterns are very hard to distinguish.
Yeah.
What about the order of them?
Yeah. And I think it turns out that the only thing that is significant, let's see, is between heterosexual men and heterosexual women.
So you're plotting and telling me
heterosexual women and homosexual women, but not heterosexual men and homosexual men. Okay. It's hard to figure it out. Okay. And so, yeah, badly presented. A not very interesting piece of research that got published in Nature.
The order of the bars doesn't allow for easy comparison.
There we go.
Yeah.
Very
People are asking about what the numbers are. Those are sample sizes. So that's actually something often to think about including, and we were just able to criticize it by saying we've collected about as much data in this class.
Yeah. Yeah, exactly. Okay. So, bad study, badly presented, and yet still highly valued in the scientific community. I hope we've made a point here. So when we take a look at some of the feedback that came out as a result of publishing this paper, you can take a look and read some of it. Of course, it's all very diplomatically stated because these are by scientific societies and groups of, scientists that are trying to be but
even in that context where scientific disagreements are often published as a very like tut tut tut, this is some nasty terminology. A house of cards built on an unknown and uncertain. The idea that only one number, it's like you're heavily into Tolkien. It's like 1 ratio to rule them all, 1 ratio to bind them.
Yeah. But Mark Breedlove doubles down, right? And he's like, I don't know a more accurate biomarker for prenatal androgen that can be readily measured in adults. So there's something there, potentially, right? It's talking about exposure in the womb to the hormonal environment that has maybe an effect, or at least is correlated with this finger measurement. Right? So what does that mean? How do we, does it matter? Do we care? It's a thing, but like, so what? Who knows, right?
Yeah.
Does it have any implication, effect on any of us as people? It's not like we're going to put like our finger length in our dating profiles. Like, oh, we totally should.
That would be funny.
Okay. So, it's a thing, but does it matter?
And how does it affect pull my finger?
Sorry. So, what does all of this mean? So, and we'll do this quite a bit in class. We'll like potentially go on these seeming tangents, but they're not. They're really important. But we want to just show you how it connects to, the human world. And we want you to then take that stuff and to apply it into freshwater mussels if you're interested in going into biology and being biologists or ecologists or veterinarians or whatever it is that you're, thinking about, to critically be able to evaluate data, to be able to transfer these ideas into the natural world.
And to think usefully, particularly about variation. I think as a criticism of lots of training of health sciences, the biological health sciences, is that inevitably with the amount of material you have to cover, just understanding our own species, if it's in the human health field or in the veterinary sciences with a handful of agriculturally important, critically important species, you end up thinking about averages and often being taught about averages. And so frequently, the average is not what you're interested in. It's the variation.
That's right. We often, thank you. In my work, I often deal a lot with something called a Likert scale. I'm supposed to pronounce it Likert, but I just can't.
You get right Likert.
Yeah.
I remember that scale from growing up.
So you've done this before, like in surveys and stuff, right? When you're asked to strongly agree, neutral, disagree, strongly disagree, right? These are discrete bars where you're asked to just click one, right? Like, how do you feel about ice cream? And you're like, strongly agree, right? Or strongly disagree. Okay. The way that people analyze those data can be very different. And in fact, there has been demonstrated bias associated with simply the analysis and who's analyzing the data, right? You can get a different result, which is bananas to me that you can, but for sure, right? If you have different approaches. A lot of people take those strongly agree, those like discrete bars on a bar graph, right? And they create an average, and they say, well, the average liking of ice cream is a 3 out of 5. But they don't understand. So if you take an average of strongly agree and you take an average of strongly disagree, you get a neutral response. So instead of seeing this binary, distribution,
it's like
people are pro ice cream,
yeah,
but they're not. Half of them are anti ice cream. And it's really important to understand the way that it's distributed. Gabriella, I think, is going to make us a little demonstration of what I was just saying, which is lovely. Oh, no, they stopped.
Talking about polling data, Hodgkin V in the chat talked about if they polled all first years from all local universities, Guelph students would know the most about mussels.
There we go.
That would be a unidirectional,
Exactly.
Mussels are life.
Mussels are life. Good. Okay, cool.
Do they secretly have fingers?
Thank you so much for today. I had a really good time.
One question. So did I. And I'm thank you. And
Thank
I'm very glad to be back and to be with you all. Difference between intra and conspecific? Nothing really. In this context, they would be used as synonyms.
Yeah.
For intra
and
conspecific.
Yeah. Vocabulary is important, right? Okay. So like when you see a question that says interspecific variation, that has to mean something different, right? So just kind of, we're always drilling down and trying to be as precise as possible in the way that we speak. So sometimes we mess up, and you're more than welcome to like ask about it. It's totally fine. But our goal is to always be as precise as possible. Okay. Are we good?
I think we're great.
Okay. Smith is going to go pick up our children.
Tips on note-taking. We have 1 more minute. Tips on note-taking in this class.
Yeah, good luck with that.
Stay with us, I would say, is my suggestion. Instead of trying to take down things verbatim.
Yeah.
Maybe if you have kind of 10 minutes at the end of class, hang around, because we stay open for the office hours.
Yeah.
But think about summarizing like the gestalt. What were the main things we talked about? Not verbatim. Because that's not really the point. It's not the point in this class. As you will learn, it's not really, or it ought not to be the point in your other classes.
So what would be a good example? We will not ask you what the average height of humans is.
Yeah, that's a great example. Yeah.
We might ask you to look at a graph and come up with 2 conclusions. So everything that we do in class is kind of studying and prepping for, well, I hope for life, but like certainly for the types of questions that we're going to ask on the topic tests, right? We will never ask you how many species of mussels there are in the Great Lakes. But certainly we might give you a scenario where there's something going on with mussels, and we might ask you to, to tell us what conclusions we can make, or what hypotheses, or what evolutionary mechanism this is associated with, that kind of thing. And we're going to give you lots of examples. So what we did today in class is an example of data interpretation that we would hope that you would practice and be able to do on a topic test. Okay. And with that,
cool.
Thank you. And stick around if you want to talk to us.
Yeah, take care of each other.